Hello, my name is Sophia Harvey, and today I will be discussing the Cree-Lock system and how it's used to knock genes out. So, what is the Cree-Lock system? Well, it's a genetic engineering tool that researchers use to delete a particular gene of interest. Basically, it knocks genes out. So, let's take a quick trip back to BIOL101 before we proceed. You'll recall that DNA is made up of thousands of genes, each of which codes for a particular protein. And proteins do useful things in your body, and they're basically what make you you. They give you all your special characteristics. When you don't have a particular gene, you don't get that particular protein and thus that particular characteristic. Alright, back to Cree-Locks. Why would researchers want to knock genes out? Well, sometimes the best way to determine what a gene does, or rather what it codes for, is to see what happens when you get rid of it. So researchers will often take mice and delete a gene of interest, creating what's called a knockout mouse, and then they'll compare it with mice that still have that gene. So for example, researchers will knock out a gene they suspect might play a role in weight regulation, and then they'll compare the weight of knockout mouse to regular mouse, as you can see here on the graph. Mice are often used because they're model organisms. They're easy to breed and house, and they have a lot of the same genes and physiology as us. So in the context of the paper that I reviewed, researchers knocked out the NEUROG3 gene, which coded for a protein that told a cell to become an enteroendocrine cell. And enteroendocrine cells are hormone-producing cells that you'll find in the small intestines. And so without the NEUROG3 gene, stem cells, or undifferentiated cells, never got the message to become enteroendocrine cells. Alright, so how do researchers actually knock out a gene? Well, they don't just go in and remove the gene with their fingers, because that would be really hard and probably wouldn't work, since genes are really tiny. So that's where Cree-Locks comes in. And Cree-Locks is all about a special DNA sequence, or rather DNA sequences, being recognized by a special protein that likes to cut DNA out. So there's two major components: LOXP sites, which are short DNA sequences shown here by the triangles, and they flank the gene of interest, for example, the NEUROG3 gene. Then there's Cree recombinase, and that's an enzyme, or a protein, that recognizes those LOXP sites and then will mediate the excision of the gene of interest. Basically, it cuts out the LOXP sites in the gene of interest. And researchers can get kind of fancy with this because they can control when they knock out the gene. So tamoxifen is a drug that activates Cree. So maybe when the mouse is like 8 weeks old, they'll inject tamoxifen into the mouse, and that will activate Cree to then go recognize those sites and cut out the gene. So how do you get a mouse with Cree and LOXP sites? Well, researchers will often breed a mouse that has the Cree recombinase gene with a mouse that has the LOXP sites flanking the gene of interest. And then they'll get a baby that has both of those components. So here we have the Cree gene, which then codes for and translates into the Cree recombinase protein. Then we have tamoxifen, which is administered, and that activates Cree. And then Cree goes, and it recognizes those LOXP sites. It then cuts out the LOXP sites in the gene of interest, And now that baby doesn't express the neurog3 gene, and it will not have enteroendocrine cells, which the researchers in my paper saw was a huge issue. Congratulations! In just about 5 minutes, you've learned about a really cool and really important research tool that biologists use all the time. I hope you enjoyed. Here's my references.